due to the sort of a potential health concern of the red dye, the FDA is trying to phase out these synthetic dyes, such as red number 3.
In our cases, by making this red soybean, we could extract this betalains, and then we could provide this natural pigments without like increasing cost or causing any supply issues, because it will have a lot of betalain amounts in the soybean.
We produced soybean seeds that had so much pigment in them that they were well above the amount of pigment that are found in red beets, like astonishingly much more. But the red color definitely goes with the Wisconsin Badger theme, right? Yeah.
There are many red colors in nature, and therefore, in many of our food, this red color is added. But in the past 50 years or so, the dominant red colors come from synthetic dye that is made out of the petroleum. So now we have to come up with an alternative, the color to add to our food. And good news is that there are alternatives, the natural alternatives, such as the beet extract or beet juice extracts. But the major problem is that it costs a lot more to produce this color, estimated to be roughly 10 times more expensive than the synthetic dye. Our lab has been interested in plant chemical pathway or plant metabolism. When we learn about this sort of the issues of or the challenges of producing natural color in an affordable and scalable manner, then we thought we can apply our expertise to help address this societal problem.
Beets can produce this red pigments called betalains, which is already used as a natural food dye. And what we did was we took this betalain producing pathway and then put them into the soybean. We also made the soybean plants to build more this building blocks needed for this betalain pigments. So we basically give our soybean plants more ingredients and better tools to produce this betalain pigments. Basically, we tested 3 different species, right? Arabidopsis, which is a model plant, and tobacco, and soybean. And then we found out the soybean worked better because the soybean has additional tyrosine biosynthetic pathway, so they're more tolerant to having to have that high levels of tyrosine in nature. That's why we think that soybean worked the best out of those 3 species. So growing part are mostly done in this WCIC facility, and then what I am doing is taking the soybean samples and then extract the metabolites out of it.
So Jung and Hiroshi had brought their DNA molecules over, and we were putting those in plants, and we were seeing results from those as well. It became really clear that it would be really interesting to rearrange the order of the genes and see if that might pay a benefit. Our process allows us to transform and put those genes into those different cultivars. And that's a really important aspect of all of this. And that's not something that everyone can do. And that is something that distinguishes the WCIC from perhaps other transformation facilities. The potential to really ramp this up in soybean, it's real, right? That we haven't hit the limit. And so that's where Soyoung's work is so important, because to make that a sustainable increase that doesn't, negatively impact the health of the plant or the ability of the seeds to germinate, those are critically important for producing a commercial product.
Our soybean actually produced a lot of red pigment betalains, but also other chemicals within those pathways. pathways, which includes like a lot of different chemicals that could be also useful for other pharmaceutical usages. So in our soybean, because we are producing these chemicals in all of those leaves and seeds and all of those tissues, so they're pretty abundant in everywhere in our soybean. We're hoping to work with potential industry partners to do more like pilot scale experiments, and then see if to figure out like how this can really fit into a real production system or platform for the soybean.
This project has been supported by USDA over the last almost 10 years, starting from very basic research. So we really appreciate the support by the USDA over the last 10 years for these projects.
Hi, I'm Emily Bauer, Director of Licensing with WARF. If you like what you've heard and want to learn more about this high potential technology, please get in touch using the information on your screen and in the description below. And don't forget to subscribe for more great videos from WARF.